All right, y'all. I'm reading a PDF that I got from my big brother Ben Judah over there in the UK, and this beautiful picture that is accompanied by the article. I will do my best to place it in the video. However, it is labeled "Science." 300 million letters of DNA are missing from the human genome. The world's most famous genetic tool has a major diversity problem. Sarah Zhang, November 28, 2018. And says here in March 1997, a man answered an ad in the Buffalo News. He agreed to give 50 milliliters of blood. He did not leave a name, as the researchers who took his blood wanted to keep things anonymous. They called him RP11. The ad RP11 answered turned out to be for the Human Genome Project, and it happened that, as the race to complete the first human genome heated up, it was RP11 sample that was ready to be sequenced first. Ultimately, 70% of the Human Genome Project's DNA came from RP11. The result came from about 50 other volunteers. Since then, one moment. Since then, the first human genome has continually refined, has the first human genome has been continually refined. It's become a reference genome, the standard against which practically every human whose DNA has been sequenced is compared. But most of it still comes from RP11. Every person's genetic code is unique, so using just one reference code genome, most of it from one person, to stand in for all humanity has introduced subtle biases into genetics research. A new study of DNA from people of African descent shows just how much the reference genome is missing. Scientists found that across 910 people in their study, 300 million letters of DNA that are not in the reference genome. Some of these newfound segments of DNA could represent new genes that were previously overlooked. Africans collectively are far more genetically diverse than people in other parts of the world, so their DNA is especially likely to differ from the reference genome. Currently, DNA sequencers work by chopping up a genome into segments that are individually read, then assembled like a jigsaw puzzle. Algorithms use the reference genome to tell them where to put each segment. If a new segment pops up, one that doesn't match anything in the reference genome, the algorithms don't know what to do. Usually, scientists ignore it. We wanted, this is in quotes, we wanted to look at what's in the pieces we're ignoring, says Rachel Sherman, a computational biologist at Johns Hopkins University, who is the lead author of the study. Sherman's advisor, Steve Salzberg, collaborates with the researchers studying genetics of asthma in people of African descent. The study includes people living in West Africa, North America, South America, and the Caribbean. It occurred to me we had this very genetically diverse group, says Salzberg. In other words, their research presented an opportunity to study the diversity missing from the reference genome. Smaller studies have found enough novel DNA sequences that they estimated there are as many as 40 million letters that can appear in the human genome, but are not in the current reference genome. Sherman and Salzberg found nearly 300 million missing letters and 125,715 DNA segments. Much more than they expected. I thought I must have done something wrong, says Sherman. But when she went back and combined combed through all her code, she couldn't find any errors. Now the question is what those 300 million previously overlooked letters of DNA contain. Perhaps they code for novel, interesting mutations related to the disease that studies using the reference genome overlooked. And they're talking about asthma and other things. Really, they're talking about a lot of deeper stuff, but I'm not going to delve off into that right now. I'll delve off into that another day. Give me one moment. Let me read that again. Now the question is that what those 300 million previously overlooked letters of DNA contain. Perhaps they code for novel, interesting mutations related to disease that studies using the reference genome overlooked. If you're only comparing against the reference genome, you're never going to find the missing pieces, says Tina Graves Lindsay, a geneticist at Washington University of St. Louis. This is especially true in people of African descent, as their high genetic diversity means their DNA is more likely not to match up to the reference genome. RP11, scientists later surmise, was probably African American himself. But the problem of using one reference genome to represent the whole human population still holds true. Graves Lindsay says that she wasn't surprised by the number of DNA sequences. Listen, he probably wasn't African American because there would be a lot larger diversity to hold it up against, and it would match the levels of diversity that Africa holds the largest level of diversity because of all the blood mixing that's gone on and because all those people are not genetically the same. However, this person was probably not of African American descent. Graves Lindsay says she wasn't surprised by the number of new DNA sequences Sherman found, as she and her collaborators are also working to address the gaps in reference genome. They're assembling the reference genomes from a diverse group of people that scientists can compare against more than a single current version. The team has sequenced 15 samples so far, including 5 from Africa. It was a very good paper for us to read about, Graves Lindsay said of Sherman's study. It proves what we're doing is really needed. But her work is still constrained by what exists in DNA repositories, there we go, which is where they hold the banks for the DNA. For example, if she doesn't have a sample to sequence from people indigenous to Australia, Since the Human Genome Project began, scientists have slowly realized they underestimated human genetic diversity. At the time, they focused on a single letter mutation, but it's becoming clear that now that big structural variations, thousands of letters being inserted or deleted or flipped around, are common too, says Deanna Church, a founding member of the Genome Reference Consortium and a scientist at the company 10X Genomics. It's like comparing 2 copies of a book to look for typos and realizing whole pages are missing from one. The big chunks of DNA that Sherman and Salzberg found missing in the reference genome are likely the product of thousands of insertions and deletions. RP11 has its own unique pattern of them, as do all other original volunteers for the Human Genome Project, and so does everyone else in the world. That's the end of the article. And give you my quick thoughts about this. How I know it's not a black person. I told you guys before in that one gene study that genetic reference studies only pretty much benefit white people, right? So why would the reference genome be not a white person? I want y'all to really think about that. There's 50 volunteers. They picked the person that they wanted to refer all genetics to. They assumed that white people were who everybody came from, probably still back then. Found out the hard way that they're not. However, don't worry. I'm going all in. I don't know what to tell y'all. I mean, I don't know what to tell you. Ben Judah sent this to me because he said some stuff that, that I was talking about, not just in the videos, but, what we were having conversations about, like, you guys got to realize, I go all in on this stuff. I live for this. Literally. I literally live for this. So I can't wait till everything falls together just a little more, because it's coming. But as I prove that these Middle Earth things were black, and I prove that they made it in with other so-called African people. See, because you see why they pull the African descendancy? They want to always call us Africans. Always. We can never be anything else to them. You see how we're still denigrated to being one of those things? So, and no offense to Africans, I'm not African. Right on? I'm an American-born black person. And even more than that, I don't even call myself an American. I call myself one by reference because that's what people know me as when they hear me talk. But I don't identify with being an American, no more than I identify with being African. So, with that said, I want y'all to start thinking, looking a little closer at the people's hair, their facial features, their facial hair features. Everything matters. People have been bred out, sure, but a lot of people haven't been bred out. So we have to look at what we have still to look at, and not by what we think is right or what we believe is right. All what is written is what we have to go by. These people are not perfect. At no time do any of these so-called geneticists ever talk about Gregor Mendel, because he was a Bible thumper. And that's the one thing that they can't have diminishing the so-called foundation of science. My people, I pray that y'all are listening. I pray that y'all are really taking heed to what I'm laying down. If not, you'll find out the hard way, the wrong way. And there won't be any time to go back and save yourself or anybody else that's with you. They'll all suffer the same fate. With that said, I'm out. I salute you. May everything that you ask for that is righteous be granted, and everything that is unrighteous be withheld.